सर लाइसोसोम जानते होंगे आप लोग। And the lysosome, basically it is known as protector of the cell, or it is also called as first line of defense. And in case of protozoans, the lysosome is known to have a stomach of the cell, because we know the protozoans engulf as the foods and endocytose as through the plasma membrane, where it makes the food vacuoles, and the food vacuoles as a fuse with the lysosome and also make phagolysosome, and as a digestion also takes place there. And that's why the lysosome has important role in also digestion in all kinds of the eukaryotic as a cell. But in case of higher vertebrate mammals, the role of the lysosome is more important, particularly in protecting the tissue or organ against the pathogen. The pathogen also that enters the higher vertebrate, including human also beings, through also different routes, either air, water, or also food. So then also it causes diseases, but however, the immunological cells, particularly the leukocytes and the cell that has processes of phagocytosis, and that neutralize the pathogens. That's why lysosome has an immunological has a role. One by one we will come. Third one has in case of insects and amphibians. And insects and amphibians during the embryonic immunological processes, they have the processes of metamorphosis. So during a metamorphosis, lysosome play an important role, because the lysosome has to contain an enzyme known as cathepsin, and that cathepsin, it is a protease enzyme that degrades the extra has a body has a component like has a tail and others, and so finally the embryo transform into adults. That's why lysosome releases contains some enzymes that helps that play an important role during the metamorphosis. Secondly, in case of higher vertebrates or during embryological process, another functions of the lysosome, that is the during the fertilization. And all we know that the sperm has to contain the acrosome, and acrosome is formed from the Golgi body, and so that contain an enzyme that is known as hyaluronidase. And the hyaluronidase, this is the enzyme that digests the fluid content or organic content of the zona pellucida and make the way through which the sperm nucleus approaches the egg membrane. And finally also it has to degrade the hyaluronic acid of that has a course around the egg membrane. That's why the hyaluronidase enzyme that has to digest the extracellular as a component of the ova and make the way for the entry of approaching the sperm nucleus to the egg membrane. In other case, another function that will be further discussed later. So that is the in endocrine, that is the release of the hormones. And all we know the name of the thyroid gland, that is known as T3 or T4, that is as a triiodothyronine or as a tetraiodothyronine, and the tetraiodothyronine commonly known as thyroxine hormone. And these are two hormones are synthesized in the parafollicular cells of the thyroid gland, and as the cytoplasm and these are hormones present, they remain conjugated with a specialized protein of the thyroid gland that is known as thyroglobulin. And the thyroglobulin has containing the T3 and T4 is further processed into the lysosome. A lysosome has to release as to the T3 and T4 from the thyroglobulin, and finally as of the T3 and T4 release as of from the cells for as of further actions. So in another as of cases as of the lysosome, that mostly as it is a vesicle that contain the hydrolytic enzyme, and the that's why as to there is any deficiency of the enzyme not synthesized in acid, that not present in the lysosome and not get stored in the lysosome, then it leads to the autosomal recessive disorder, that is also known as lysosomal storage disease. And another point now has to become very fast the organizations of the lysosome. All we know, the lysosome is present in eukaryotic cells, including protozoans, few plant cells, and as animal cells. And higher as a vertebrate or other eukaryotic animals, they have large number of the lysosome. But in mammals, the number of the lysosome is more than that of the other animals. Secondly, as it was when it was studied as by the Duba during as of 1949 onwards, and it was studied as a vesicle that was an intermediate in between as a mitochondria and endosome. That then as he observed a vesicle containing hydrolases, mostly acid phosphatases. Later, and this acid phosphatases that is involved in the digestion of the organic biomolecules, and later one it was named as lysosome, and he also observed the activity of the lysosome in pathogenic conditions. So then it was known as the lysosome that contain the hydrolases, or whenever also it conjugates also with the pathogens, bacteria or other as a component. So then also it also kills the pathogenic pathogens, and however during also that digestive processes or degradative processes, the cell get also also killed. Sometimes this lysosome is also known as suicidal bag has, but however the suicidal tendency is not how because the lysosome play an important role in several biological functions, physiological as of functions, including they have the immunological role in release of the hormones and others and others. Anyway, of the lysosome, that is it is a vesicle, single membrane has a vesicle, and it contains as these so many kinds of hydrolytic enzymes. And also one also it's the membrane. The membrane has a composition that reflects the differences in between other cell organelle, a cell organelle with others, because it has a vesicle that has acidic pH. At this pH that fluctuates, the lysosomal pH fluctuates in between 4 and 5, sometimes as 4.5 and as 5 others. And so this acidic pH, this is due to the presence of HPT and proton ATPase. This is a proton ATPase that is the important as a membrane as a protein that act as a proton has a pump. And this as a proton has a pump. It is in the ATPase enzyme, and as the incorporation of it has accumulates the ATPase. It accumulates the as a proton. This as a proton as that is produced inside the cell, and during the metabolic processes or respiratory process, and another cases CO2 combines with the water and also produce H2CO3, and this H2CO3 then also dissociates into proton and bicarbonate H2CO3, and this also further dissociates in the proton, and this also surplus as a proton as also the proton concentration increase in the cell, Then so this proton is accumulated in the lysosomal membrane, lysosome, so that lysosome very first protect the cell from their acidic in nature, because the general pH of the cell cytosol of the pH is 7.2. And if the proton concentration increases inside the cell due to metabolic processes, or as even though concentration of carbon dioxide increases inside the cytosol, this carbon dioxide react with H2 and then H2CO3 or H2SO dissociates into proton and others. Whenever if a pH of the cytosol has decreases from the 6, then it has detrimental effect on the cytoplasm, yes, cytosol, because in as a low pH, acidic as a pH, enzymes are non as a functional, and other as a consequences also as a takes place. The protein is also become as a, yeah, does not function very well. That's why they create the consequences in the cell. The second one, the importance of the the lysosome that accumulate the proton inside the it. That's why very first role for the cell has to and protect the cell is the accumulations of the proton inside it. That's why someone in a lysosome called as protector of the cell, or as the, because it has to protect the cell from so many kinds. Another reason it is also called as first line of defense. First line of defense, that is the it has immunological role, etc. Second, the membrane composition. That if you look at the membrane composition, then it has to contain the phospholipid and a sphingolipid, and the concentration of a sphingolipid little bit is higher in comparison to other membranes. Secondly, it also the characteristic features of the lysosome having the phospholipid is, then it is contain the bis monoglycerol phosphatidides, bis monophosphoglycerol, because the glycerol that contain one molecule of fatty acids rather than other phospholipids that is present in the membrane that has nearly two fatty acids. But the membrane phospholipid that constitute the membrane, lysosomal membrane, that contains the one fatty acid, and at the last carbon it contains the two phosphates. That's why it is known as bis monophosphatidylphosphatidyl phosphatidides. Second, the inophosphatidides, yeah, 1,3 bis phosphatidyl glycerol, phospholipid, inositol, glycerol, phosphate, phosphatidyl, and yes, you can say, yes, so glycophospholipid that has contain one molecule of inositol and also three molecules of phosphate. That is also present in the lysosomal membrane. And the protein content is also less in comparison to others, because other membrane that has characteristic features to having the certain enzymatic activity, and so they also have receptor and others a transport system. But the lysosome that does not do not contain receptor and others, but few receptors are present, or only you can say the receptor has the well-known receptor present in the lysosome is mannose-6 phosphate receptor. Mannose-6 phosphate receptor is, that is the indicator to identify the lysosome. Any membrane has, if a cell undergoes homogenization process, then how we can study as which membrane is which component belongs to which membrane, which has organelle. So then one objective is that then this is the Gumori stain, Gumori stain. The Gumori stain that is used for staining the lysosome, and it is also basically this Gumori stain that is the used also for histochemical usage, and also the Gumori stain that stain the acid phosphatase, and also the presence of the acid phosphatase mark the identifications of the lysosomal membrane. That's why Gumori stain is well known stain as a for studying as the lysosomal as a membrane. Second as objective, that is the uh, one, so that is the enzyme that contain on the membrane, that is proton ATPase. Second is the mannose 6 phosphate receptor, the 6 mannose 6 phosphatase. This is present on the lysosomal membrane. However, as one thing that is basically the higher concentration or acidic has a pH. Question is how, because sometimes some writer say because it contains the hydrolytic enzyme, or whenever lysosomal membrane destabilize, that then it releases the enzymes that is acidic in nature, and as it act upon the organic molecule, sometimes it causes the killing of the cell. But still, there is not a lot of the enzyme as present in the lysosome that can hydrolyze as all as the cellular component. Anyway, but so sometimes, yeah, whenever such condition appear, then it kills the hazo cell, or this phenomena is known as autophagy, but or also called as autolysis. The lysosome is known as for having the autolytic as a process or autophagy processes. But otherwise, it is known as protector of the cell as because the higher concentration of proton that is accumulated inside the lysosome, and the enzyme that is accumulated are present in the lysosome, that functions in acidic phase. Up second, now as of the enzymatic compositions of the lysosome, if we look at, then also it has been reported as that there is about 50 enzymes, and the that is present in on all of the 50 enzymes belong to hydrolases, because there is 6 classes of enzyme: oxidoreductase, transferase, and third as a class of the enzyme that is known as hydrolase. Fourth one lyase, no, and पांचवा the isomerase, and last one the ligase. So that is the class third of the enzyme that is known as hydrolase. And hydrolase, it is an enzyme so that split the covalent bond, and the covalent is a bond so that split, that include the enzyme so that split the ester bond, then so it is known as esterase. So that is the naming of the enzyme so that is conjugated with the substrate and is ending with the ASE. So then so if has a enzyme present in the lysosome that act upon the ester bond, ether bond, peptide bond, and peptide bond is basically it is called as protease, glucosidases, phosphatases, nucleosidases, nucleotidases, and this nucleotidases and nucleosidases, and another enzyme that is associated with the nucleotidases like ribonucleotidases, deoxyribonucleotidases, and so the other hyaluronidases, acid phosphatases, and so many enzymes that is glucuronidases. This enzyme is present in the lysosomal lysosome that is enclosed by the single HSO membrane. That is single HSO membrane. The basic HSO, the enzymatic compositions of the lysosome is that has been classified on the basis of they bond, they split the chemical HSO bond, covalent HSO bond that is present. That is present in the macromolecules or microbiomolecules. Micromolecules like as a DNA, RNA, so proteins, etc. And microbiomolecules like as the oligosaccharides, disaccharides, tetrasaccharides, peptides, and etc. It means as basically micromolecules means the biomolecules that contain nearly about 25 carbon, but as a macrobiomolecules is known about because as they have the carbon content higher than that of the 25 carbon. This is the basic differences in between microbiomolecules and macrobiomolecules. That the lysosome so that hydrolyzes basically on the bonds like glucose added, that is uh, in between so two glucose, yeah, during the in a starch, cellulose, and etc. That is known as glucosidic bond. And in case of the polypeptides and protein, they have as a peptide as a bond. In case of the nucleic acid, they have 3 or 4 more kinds of the bonds, like as a phosphoester bond, phosphodiester as a bonds, and then the glucosidic, beta alanine glucosidic bonds, the such kinds of the bonds also present. Some other as food biomolecules also contain the sulfur. The sulfuryl, when the sulfuric acetate, when sulfuric hydrolysis, the sulfuryl hydrolysis that is also present because it present in the lysosome because it hydrolyzes the thioester as a bond, thioester bond, that is the thio means sulfur or sulfur as that is there is two amino acids and that has the sulfur in form of SH sulfur hydryl group like cysteine and methionine, but however cysteine play an important role in making the disulfide bond, and also it has some it conjugate with the other non-protein also, that is the acetyl CoNAME, cyl CoNAME, and others. Then it also split the sulfurate and other enzyme thioesterase that hydrolyzes the ester bond, so that is made in between sulfur disulfide bond or thioester as a bond with the protein with another has some molecules, by in case of coenzyme vitamins खास करके B5 that is also known as ये क्या उसको बोलते हैं coenzyme A. Second also in and the another characteristic features of the lysosomal enzyme is because it has contain it contains also the enzymes, they also they are glycoprotein in the nature, mostly as they are glycoprotein in the nature. And another objective is that the hydrolytic enzyme that has amino acids in their active sites having OH group like serine or threonine. Sometimes the other the amino acids that is involved in oxidation reduction reaction, but however lysosome also contain varies. That is also known as serine protease or threonine protease. Mostly the lysosome contain the hydrolase belongs to having the enzyme that contain the serine or threonine in their active sites and other mostly because this amino acid evolved in due course of evolution and for having the hydrolytic as a function because it has split the covalent bond in between as a monomeric unit like amino acid, yeah, the sugar, etc. One has a question generally because the lysosomal membrane that has less concentration of cholesterol, or it is studied by applying the antibiotic. Now there is I am applying three antibiotics. One that is the tunicamycin, and this is the tunicamycin. Whenever cell is treated with the tunicamycin, then it inhibits the glycosidations of this protein. Glycosidation simply means the glycoprotein that is being synthesized in the rough endoplasmic reticulum, and from RER it is further carried to the Golgi body, and as Golgi body where streaming and further modifications takes place, that is also known as glycosidations, and the tunicamycin inhibit the incorporations of very first N-style glucosamine phosphate. If N-style glucosamine phosphate that is inhibited, then the further accumulations of the lysosomal enzymes is further synthesis of the lysosomal enzyme is inhibited, and then the lysosome unable to incorporate the enzymes. Second second antibiotic that the puromycin, and puromycin is also that inhibit the glycosylation and translations, particularly the aminosylation. Then if the cell is treated with the puromycin, then it inhibit the translations of the proteins. That's why any protozoan or bacteria, खास करके if as a cell is infected, people are infected with the bacteria, then puromycin is applying, then it inhibit the associations of the ribosomes, but particularly a smaller subunit or larger subunit, and the ribosome that also provide the attachment of the amino acid transfer RNA. That's why it inhibit and interfere the attachment of the amino acid transfer RNA to the ribosomes. That's why it also inhibit the synthesis of the protein. That as a result, the bacterial cell get killed, are unable to synthesize the protein for their metabolic activity. That's why it is used for treatment. Third, that is I use that is there's two objective. In case of malarial parasite or malarious diseases, the quinine or other amines or a quinine that contain the amines. And whenever amines, when a quinine, so that is basically used for treating the malaria. That this is a quinine that contain the amine, and this amine incorporates in the lysosomal membrane. And as it comes into it enters the lysosome. So then the amine quinine that contain the amine that raise the pH. That's where the pH raised from 5 to 7 or 6 or 7. And as a pH of the lysosomal as a protozoans when lysosomal increase, that then it does not have functions as a hydrolytic center, because as a protozoa that contain as a lysosome that act as a stomach, primary stomach, and as their digestion takes place, primary digestion takes place of the food stuff. That it has to increase the pH because amine increases the pH, and as it has a pH increases. Another objective of the lysosomal membrane is that if pH range from 4 to 5, then membrane is stabilized. However, if pH further increase from 5 to 7, the membrane is destabilized. During that period, the membrane lysosome membranes of the lysosome get ruptured and releases the enzyme, and that enzyme further engulf the substances that is present in the protozoan, and parasite get killed. So that the amine quinine, there is role of the three. Tunicamycin, that is antibiotic, puromycin is antibiotic, and quinine that is applying for treating the malarial parasite that contain the amine, or whenever amine is applying to the infected peoples, that then the amine enters the lysosomes of the pathogenic cells. And even because malarial parasite is also eukaryotes, and human beings cells are also eukaryote, that's why it also have some negative role, a physiological role on the host also. But however, very first we have to protect ourselves from the pathogens. That's why very first treatment that is given to the malarial patients. That is the quinine that contain the amine, or amine that raise the pH, and further destabilization of the lysosomal membrane takes place. And the destabilized membrane has to release its enzyme, and then has to digest the cellular content of the parasites. Overall, so this is the basic meaning of the lysosome. Now, we have to focus about the polymorphism in the lysosome. And polymorphism is known as, what is polymorphism? One question is, polymorphism क्या है? Lysosome existing more than one form or others? So basically, there is physiological existence of the lysosome from their occurrence to the final maturity. Lysosome exists in four or more form. In general, the polymorphism, the basic meaning, the same content and the same structure exist in more than one form that differs physiologically. And physiologically means they contain the enzyme and others, and so their function is different from everyone. Then it is called as polymorphism, particularly for the lysosome. But in case of invertebrate like hydrozoa, coelenterate, so that exhibit as maximum polymorphism, where it is not applicable. So then anyway, and so the lysosome that exhibit the polymorphism is known as primary lysosome, secondary lysosome, and then has a residual body, and fourth one autophagy. That there is just a four physiological as a changes of physiological name that is associated as with the lysosome. And if you have to look as because the lysosome that contain the enzyme, the very first, the origin of the lysosomal as a protein. The lysosomal as a protein that is synthesized on the rough endoplasmic reticulum. And the rough endoplasmic reticulum that synthesizes as a protein, or after synthesis, this as a protein is packaged into vesicles. Hello? Anyone? Vesicles. And this as a vesicle as a further and carried as a to the Golgi body. And as a Golgi body as a we know later one the Golgi body that exists in also exists in as a four configurations. And these four configuration of the Golgi that is known as cis Golgi, medieval Golgi, trans Golgi, and trans Golgi network. So that has this is the the protein that is being synthesized in the rough endoplasmic reticulum, and from also the lumen it is packaged into the vesicle and budded also from the endoplasmic reticulum, and also then it is also carried also to the Golgi has a body. And as a Golgi body, all we know it is known as postman of the cell. And postman of the cell, it means it also causes the sorting of the protein. And after the sorting, the protein is carried to the respective at the respective addresses. Like as if there's a protein being synthesized as for the lysosome, or or also for the RER, also for the membrane, and also the protein that has been that has synthesized in the RER, and it is also released also into the cytosol, extra cytosolic site. That's why also the Golgi has a body also that act as a postman, because it has very first sorted the sort the protein, and after the sorting they also deliver as to the delivered for respective addresses. And that's why the lysosomal as a membrane, all these not lysosomal membrane, all the lysosomal enzyme is being synthesized in the rough endoplasmic reticulum. So that after selection, the messenger RNA is further translated and then also enters, and also it is glycosylated, and the glycosylated, yeah, the oligosaccharide present in the protein as a residue, that is the because so one objective is that like for the lysosomal as a protein, because of the oligos in last class we had discussed the protein, the oligosaccharide or sugar residue that is added to the polypeptide, a newly synthesized polypeptide in the RER that has asparagine at the end terminus, show that as the very first characteristic features of the lysosomal as a protein as to have a sugar residue that has N-linked glucosidic bond, and the L-linked glucosidic bond that is made in between a sugar residue and also newly synthesized a nascent polypeptide that has an asparagine. Very first, so the our seven objective is that pH of the endoplasmic reticulum is 7.5, and or as a Golgi as a body that has, you can imagine in case of cell that has 2 pH less than that of the RER. Some book has so that have written as the pH of the RER that is 7, but if you go to the Stryer, then it will find as the pH of the RER that is nearly what 7.5, and so the pH of the Golgi body has just as 5.5. So that has there is at a 5.5. So there is pH in between as RER and as a Golgi body, so there is 2 less pH less than that of the RER, and the pH of the lysosome that fluctuate, that depends upon the physiological conditions of the cell. If as a physiological conditions of the cell, in the sense if any pathogenic condition and others, if as a proton concentration increase in the cell, then also the pH decrease from 5 to 4. However, if pH of the lysosome that fluctuates in between 4 to 5, or So that also there is just the organelle that has pH order, and that is or if a nucleus, the pH of the nucleus nearly what 8 or 8 point something. That's why so from the nucleus then so RER then so the Golgi has a body, or if you look at the mitochondria where is pH has nearly what 6.5 or 6, never has to increase, never has to pH, they have pH nearly what the 7, even though the proton concentration is higher in case of the matrix of the mitochondria, in spite of the higher concentration of the proton in mitochondria, but so pH never decreases so below the 6. That's why you can also rearrange also the pH organelle according to the pH order. When 8 or 8.5, then 7.5, then also 6.5, 6, 5.5, then also 4 or also 5. And still it has, because it contains the proton ATPH. Another also cases that you will also find, that is the endosome. They also have lower acidic pH. So then, of the very first also identifications of the also protein and is that is also present of the mannose 6 phosphate. Mannose 6 phosphate. The also, sugar residue is added to the protein nascent protein that also contain the sugar, but they contain the two molecules N-style glucosamine, three molecules of glucose, and 9 molecules of mannose. So that 9 plus 3, 12, or 12 plus 2, 14. So that in RER there is also 14 sugar residue is present. Now, after synthesis, the glycoprotein has so comes to the Golgi body, and also the Golgi has a body has a very fast. The very as a first process mechanism, during delivery of the glycoprotein to the Golgi body from the RER. The where is also the 3 has a glucose residue, that is streaming of the glucose residue. Because the protein does not contain the glucose. However, it is added in the RER. But so at the time of the functions, the SO3 has a glucose residue must be removed. So that also, if you also go to the books, then you will find the 3 glucose residue, that is the removal of the 3 glucose molecule from the glycoprotein is a critical decision for the protein that is that has to transport for further modification in the Golgi body, and from the Golgi body then also to lysosome, a membrane, and others. The very first mechanism, the removal of, and also the several also time this 3 glucose residue is also removed and added as final decision. It means also the glucose हटते जाता ऐड होता माने glucose is removed and added so that also the several times streaming takes place. But so whenever as a cell make decision, yeah, so Golgi body make decision as for further modifications of the glycoprotein to deliver to other cell organelle or other as cellular component, then also the glucose residue is not added as of further. The very first mechanism is the removal of the 3 glucose that has been added to the RER. And so several times trimming, adding, trimming, removing, adding. That's why it is known as trimming. So that's a trimming of the glucose residue and the glycoprotein has so takes place. And another has the most important as of functions, which as an enzyme is of which. And already as in RER, there is an address. There is an address also for a specific also proteins or enzymes. Yeah, the protein that act as an enzyme is already also present on the messenger RNA that is translated, and after translation, certain amino acid residue added, and the certain amino acids that is at the end terminus that act as a signal sequences, and this signal sequences is recognized by signal recognition particle, or this signal recognition as a particle carried to the RER where it is attached to the membrane, and also further translation takes place. Or after translation, the protein undergoes glycosylation. During glycosylation, the 14 sugar residue is added, and very first sugar residue is added there. That is the N-acetyl glucosamine, and there is 2 molecules of N-acetyl glucosamine. And after 2 N-acetyl glucosamine, there is also 9 also molecules of mannose in also 2 parts are added. Later, so 3 glucose molecule is added. Now, so that's why some total of 14 sugar residue is added to the newly synthesized proteins in the RER. After also RER, so this must be also delivered to the Golgi body for further modifications. And the modification that is made in the Golgi body, and also from RER to the Golgi, there is just a trimming process takes place in so 3 glucose residue, because the protein sugar residue present on the protein does not have the glucose residue, but the mostly the protein has sugar residue that belongs to 4 or 5. That is the N-acetyl glucosamine, mannose, N-acetyl glucosamine, very first. There is second position, there is mannose. Third also position, that is the galactose, and so fourth as N-star neuraminic acid, and last one as that is the fucose. So that is so 4 or 5 as a sugar residue is added, yeah, remain as a present on the glycoprotein. So that's my very first trimming. And after as a trimming, and the mannose that is exposed to the glycoprotein, and so these mannose also does not have the phosphate group. So that very first as a UDP, and this UDP, this is the nucleotide that contains also the N-acetyl glucosamine, simply called as NAG. And the N-acetyl glucosamine, this N-acetyl glucosamine, in presence of N-acetyl glucosamine kinase, the has a phosphoryl potential, it then has to transfer to the N-acetyl glucosamine, and so that phosphorylated N-acetyl glucosamine that has to transfer its phosphoryl potential to the mannose residue. So that is a mannose. And so this mannose phosphoryl potential of phosphate is present at the 6th carbon, 6 carbon of the mannose. That is called, that's why it is known as mannose 6 phosphate. And so that is the mannose, so that is exposed, that has phosphoryl potential, that has phosphate group at the 6th carbon, not it remain present on the 3rd carbon or other as a carbon. Only as the identification of the lysosomal enzyme, that is the presence of mannose 6 phosphate. And so whenever as a phosphate is added to the mannose at the 6th positions, then it is now known as mannose 6 phosphate, and 1 or 2 जितने mannose है वहां पर ऐसे they 1 or 2 mannose residue that remain exposed, or this also 2 mannose also that accept the phosphoryl potential, and this phosphoryl potential is also transferred by the N-acetyl glucosamine UDP, then as a mannose 6 phosphate, and this mannose 6 phosphate contain the enzymes of protein that is then also transfer also to the Golgi has a body, because as a Golgi membrane, lysosome, because as the membrane of the lysosome that contain the mannose 6 phosphate receptor. Any has the phosphatase and other as destabilizing enzyme that can inhibit. Let's say, for example, if you also apply the tunicamycin, that this also tunicamycin that inhibit the very first as attachment of the N-acetyl glucosamine and UDP. That's why whenever cell is treated as with the tunicamycin, then also the phosphoryl potential is not also added to the mannose 6 phosphate. That's why also the enzymes that is present in the Golgi body that has comes into the cytosol, etc. That's why also the lysosomal enzyme is not accumulate into the lysosome. That's why it causes a disease also known as eye disease. It is known as one you will find as eye disease. And one topic यहाँ पर अगले क्लास में करेंगे, there is a known as eye disease. The eye disease, जितने eye disease है, there it is the concerned with the lysosomal. Later one, autosomal recessive disorder is another as a topic, so that is the associated with the lysosome, that is also known as lysosomal storage disorder. Anyway, I will also discuss later. The very first as the tunicamycin, the role of the tunicamycin, that is antibiotic. This tunicamycin inhibit the additions of the N-acetylglucosamine with UDP, and then that's why the mannose is not phosphorylated as of with the I do not have the 6-phosphate and its 6 carbon. That's why the entire enzyme has formed, but they are not delivered as to the lysosome, because as the receptors present on the lysosomal membrane that recognizes the enzyme, lysosomal enzyme as bearing the 6-phosphate at the mannose residue. That's very first as the clue that is for the synthesis of the lysosomal enzyme. The primary lysosome is known for vesicular lysosome, because as it is the preparatory lysosome, because as during which you can say the preparing or storage lysosome also called as it is also called, because as the enzyme it accumulates the enzyme and physiologically is not active. That's why this is the primary lysosome. The most active as a physiological role of the lysosome is the heterophagy or secondary lysosome. And the secondary lysosome that make one or two more kinds of the phagolysosomes. In case of the bacterial infection or pathogenic infection, like as the lysosome, the primary lysosome that change into the secondary lysosome, and the secondary lysosome that contain the mature enzymes, and physiologically it is active. Our second characteristic feature, because it is formed by the budding of the breakdown of the Golgi body, cysteine or aria cysteine. That's why it has pH somewhat fluctuates between 6 and 7. But the functional role of the functional pH of the lysosome must be range from 4 to 5. So that is a primary that has pH higher, primary lysosome that has higher pH like 5.5 and 6, because it is budded from the Golgi body. Then the pH must be decreased from 5.5 and that up to 4.5 or 5. So that another as a source that previously I called, because it contains the proton ATPase and proton as ATPase, that is the proton has been pump that has to accumulate import as a proton from the cytosol to the lysosome. As a result, as to increased concentration of the proton that has to decrease as a pH, and that's why decrease as a pH, because already we know, for example, if you know, because the stomach has pH 2 or nearly about 2, and this has a 2 pH is required for the primary digestion of the protein, because the peptidase pepsinogen, the pepsin enzyme that is present in the stomach, that cleave the protein into proteoses and peptones. That's why same thing happens here also, because the very first when a protein digestion and takes place in acidic environment, but still it is not clear about the lysosome, because the all these food stuffs are hydrolyzed, because you know the intestine and where digestion of takes place in alkaline as a medium, but as alkaline does not, when alkaline is not concerned with the lysosome. So that still it is it has to clear us about the lysosome that contain the enzyme that can digest all the food stuff, at least is not clear. Anyway, then there's a secondary lysosome that is also called as heterophagy phagolysosome. Then also the cell cell has also two kinds of import system. One kind that is the exocytosis, endocytosis, or endocytosis has because inward as the movement of the substances across the plasma membrane, or endocytosis has more than one kind, because what if as a cell intake the solid as a food, then it is known as phagosome or phagocytosis. Whenever cell has some ingest the liquid substances, then it is known as pinocytosis. और यहाँ पर जो endocytosis है, तो the endocytosis also है ना, it is of two types. One kind endocytosis in where membrane has to invaginate itself, है ना, invaginate. So then also during invagination, the lysosome that fuses as a with as a vesicles and make as a phagolysosome. So this is also called as phagosomes, and vacuole is formed by fusing the vesicles, that is the membranous as a vesicle that contain the pathogenic substances or other substances or solid substances, and then it has a fuses with the lysosome, then it is called as phagolysosome. Second most important as a phenomena, so that is the endocytosis, that is known as the receptor mediated endocytosis. This is the known as the receptor mediated RME. And this receptor mediated endocytosis is very well known as a phenomena for the SO cell, because the virus enters also through the enters inside the SO cell using the receptor mediated endocytosis. If cell doesn't have the receptor, that's why the cell cannot allow also the virus to get entry inside the cell. That's why so the cell infect the higher vertebrate, including human beings. So that is if the cell has well developed receptor mediated endocytosis, then virus get entry. But as like for the lysosome is concerned, the lysosome has that mediate the processes of phagocytosis generally, engulfing this solid as a components already contained. And the as a membrane as a has important role, play an important role, because it protect the as cell that and doesn't allow the substances to come in. But in case of look at the so it case protozoans, and we know protozoans and the formation of a food vacuole, and that is linked also with the two. One, that is the in case of nutrition, and also during nutrition, the protozoa also has coupled process that is for the locomotions. So that is the capturing the food, and also somebody has linked also with the locomotions, because the engulfing the food. Then also anyway any organism that may change their place from one place to another place during as of food. But however, in case of protozoa, the membrane is entirely involved for capturing the food, particularly the amoeba. Then also whenever food is also there, then also amoeba sends its pseudopodia, and so you know pseudopodia is concerned with the locomotion, and then pseudopodia slowly surround it, and so then also after surrounding, then a little bit of butter, then it is engulfed by the membrane and invaginate further into lysosome cell. And so this is known as food vacuole. This also food as a vacuole. Still, its pH is not clear depending upon condition. But however, so this as a food vacuole fuses also with the lysosome and make the phagolysosome. And whereas a primary digestion takes place, because the lysosome that contain the enzymes that split the covalent bonds, that is, this covalent bond is present in polypeptides, proteins, macromolecules, carbohydrate, lipids, and etc. That's why so it is further degraded, and as after degradations, and the lysosome then has to release its partial digested food into the cytoplasm, and further digestion in case of the protozoa takes place in a alkaline environment, just like as a human beings and other vertebrates. But in case of humans, yeah, higher vertebrate, the leukocyte or leukocytes and others, particularly the neutrophils. Neutrophil is known for having the phagocytosis, and also the WBC is because it has immunological role, and this WBC is that contain also the two characteristic features, because the leukocyte, the neutrophil that is moving along with the blood flow, and during also the blood as a flow, whenever any pathogens or other substances come in contact, then also it engulf. And as it is not the common as a process, but however it is a physiological as a process. After as engulfing, then as a membrane, because as it is also has polymorphonucleus, and the polymorphonuclear as a cell, and that is well developed lysosome, and there's a well as a developed lysosome, and that would number depend upon conditions, number increases as 10, 20, and more and more. So then the lysosome comes, or as then as the food as they are pathogen, solid waste as the harmful substances that when attaches to the lysosome WBC membrane, liquid polymorphonuclear as a cell membrane, that is the neutrophils, and this neutrophil as then membrane invaginate inside. But in case of receptor mediated endocytosis, there is a role of the protein that is known as clathrin. वहां पर आप देखेंगे clathrin के बारे में. But in case of neutrophil, some proteins है ना, that make as a membrane as a flexible, and like a spectrin, ankyrin, and other membrane as a protein that also allow this membrane to change their shape, and as a result of changing the shape, they are pathogenic or including bacteria and others, they are engulfed, or after engulfing they might make the vacuole, that is the primary vacuole, and this primary vacuole as fuses with the lysosome. Primary lysosome, sometimes books also write that it is known as primary lysosome. Whenever the vesicle vacuoles containing the pathogen fuses with the primary lysosome, then it makes the secondary lysosome. After secondary lysosome is formed, then further digestive hydrolytic processes start, or hydrolytic processes when is now over, then the pathogen and others, and now has to destabilize, or they remain as non-functional. But sometimes in case of the immunological response, the it is known that the entire polypeptides or pathogens, harmful substances of pathogens are not degraded. Later one it is handed over to the other lymphocyte cell, जिसको आप कहते हैं cell mediated or humoral mediated immunity. Later one you will see. But however, as in this cases, after digestion, then that's why the presence of the lysosome in the neutrophils, and it has to protect the body, protect the cell, protect the organ against the pathogenic conditions. That's why in that immunological response, the lysosome has been said as the first line of defense, the very first line of defense. Still, as I am a human being has synthesized so many kinds of antibiotic, but huge amounts of the animals, including as higher vertebrate, they have such reach as they mostly they face the natural pathogenic pathogens that survive in the nature, and how they have to protect, so that as nature has provided so many as protective measures in their body. As a result, as they protect themselves in a human as a body, or when a higher vertebrate also, including as higher vertebrate like as mammals, where as the lysosome play an important role and in as phagocytosis. After phagocytosis, some little bit has to include some other substances also. So then as primary functions of the primary lysosome, that is the storage lysosome, also called a storage lysosome, that store the enzymes, hydrolytic enzymes, but it is physiologically non-functional unless and until they are not fused with the any pathogen or any condition. So that it is known as secondary lysosome or heterophagy. Third condition, that is the residual body. The during digestion, some substances that is the remain purged, and as they are not digested by the lysosomal enzyme, either due to lack of the enzyme or due to the pH or physiological environment of the lysosomes. That's why they get stored into the lysosome. After storage, slowly, this is known as residual body. And this residual body, sometimes it has again moved to the plasma membrane, and from the plasma membrane, it further fuses and is released in the extracellular side. That is also called as exocytosis. So that the residual body that removes its substances through outside the cell through the processes of exocytosis. That's why lysosome also has exocytotic processes. Fourth one, that is the autophagy. Or autophagy, so in some condition, particularly the starvation or fasting, or however starvation, during a starvation or fasting, there is oxidative है ना, the cell that faces major problem against the oxygen, that is known as oxidative stress. Oxidative stress in the sense that because we know mitochondria is known as for oxidizing all kinds of the foodstuffs into carbon dioxide and water, and that is coupled with the oxidative phosphorylation. But however, as food is not as there, or food is present in the body as get now over. But as oxygen consumption is continuously as takes place, or as a result of oxygen concentration increase, then the this oxygen that enters the mitochondria, whereas there is no electrons, because oxygen is known as ultimate electron acceptor. Has oxygen is known as ultimate electron acceptor. As a result, as oxygen concentration increase in the mitochondria, then it has causes peroxidation of the mitochondria. So that whenever the peroxidation, like as it form the H2O2, on this H2O2 has the H2O2 has a negative role. It has damages the cell. That's why in during a starvation, if food is not there, food concentration decreases, and however oxygen concentration increases, then increase oxygen concentration that damages the mitochondria and cell also. In that condition, the lysosome engulf little bit of the mitochondria, and that's why it protect the cell against the peroxidation process. This process, and also it engulf the little bit of the membrane of the RER, we have endoplasmic reticulum. This phenomena is known as autophagy, or this autophagy is performed by the lysosome is another functions. Sometimes it also whenever its membrane get destabilized, or sometimes other physiological changes also takes place in pathogenic conditions. That's why it releases its enzyme, or whenever enzyme is released into the cytosol, then it digests the cytosolic substances. In that case, it causes the killing of the cell. This is known as autolysis. That there is, and also the phenomenon of phagocytosis or autolysis is very concerned also with the concerned during the metamorphosis. During metamorphosis, like in case of the insect metamorphosis, that is the ecdysis, or in case of the amphibians, that the degradations or digestions of the extra organ, like as a tail, sometimes as in case of insect, the other as a modified as a body as a parts that is not needed during the for the adult. That's why it must be as a remove. That's why the removal of extra organ body as a parts is carried out by the release of the lysosome. That's as a lysosome. That's why it is involved during the metamorphosis. And in case of the amphibian, the very well known enzyme question आता है इसमें, that is cathepsin. And this cathepsin, this as a cathepsin is the protease, and this as a protease also that digest the protease when proteins contain, and as cathepsin, and this as cathepsin and digest the tail, the extra organ tail, because the adult amphibian does not have the tail, but the tadpole also that has tail. So that metamorphosis from tadpole to as adult or young one amphibians, the tail is digested, and other as a surplus as or component, because they also had waved as feet, and this is also digested with the protease released from the cathepsins. Second, another objective is for the humans, so that whenever pregnant mother deliver a baby, the when when a baby is matured and ready for protrusion, then also the size of the uterus increase and weight of the side uterus is also become the 2 kg. Normal size of the uterus is the 50 gram. So normal weight of the uterus is 50 gram. And this has a 50 gram. And during as a fully mature uterus that increases large size, and also the weight of the uterus that also increases from 50 gram to 2 kg. So after delivery, so these extra large uterus must be resume the normal weight, that is the 50 gram. So that after parturition within 9 days, the uterus when a uterus that is digested as with the lysosomal enzyme. So that again as it is known as the it is called as a process of the autolysis, because lysosome has an important role as during the restructuring of the uterus from 2 kg as of bed to 50 as of grams, so that as these extra cells are dead, that get killed or as a diet, or as another like another point that is the it also remove the as debris of the uterus as a cell or cell. As a result, as the uterus resume its normal as a size. So that this is the or less as the functions of the lysosome, that is the lysosomal diseases, eye diseases, then one things that is the immunological role, और ये स्टोरेज डिसऑर्डर जो इसको हम कराना है इन लास्ट नेक्स्ट क्लास आई विल डिस्कस विथ यू एनी क्वेश्चन इफ यू एनी हैव एनी पार्टिसिपेंट
No, sir.
यस यस यू कैन आस्क एनीवन
सर टाइम इज 8:10 तो क्लास लीव कर सकते हैं आप?
हाँ बेटा
सर 8:10 हो रहे हैं, क्लास लीव कर दे क्या?
हाँ लीव अब नहीं है, मुझे तो कह रहे हैं कि तो कुछ क्या हुआ प्रॉब्लम है, अगर क्वेश्चन होगा तो बता देना, फिर डिस्कस कर लेंगे। जो क्या नाम है आपका?
सर सोफर।
है
सोफर।
अच्छा अच्छा है ना, ठीक है। तो ठीक है, आप लीव कर देते हैं बेटा, लीव कर देते, ठीक है। मैडम नमस्ते। हम हाँ हो गया बेटा, अभी ऐसे इसका ये वाला क्लास था, नेट का था, अभी जस्ट ऑफ कर रहे हैं। हाँ कर दो, बताए मैडम। हाँ हाँ।
मैं